Eukaryotic and prokaryotic cell functions. All living organisms are made from cells which can be prokaryotic or eukaryotic. Bacteria are made of prokaryotic cells which have less organelles as they're much smaller. Animal and plants are made of eukaryotic cells. Prokaryotic cells are smaller than eukaryotic cells. Cells are made of organelles, and organelles have specific functions. Animal and plant cells both contain these organelles. 1. Nucleus, which has DNA that codes for specific proteins to build new cells, and the DNA is enclosed in a nuclear membrane. 2. Cytoplasm is a liquid substance where chemical reactions happen in. Enzymes are in the cytoplasm, and enzymes are biological catalysts made of proteins that speed up the rate of reaction without being used up. 3. Cell membranes control what enters and exits the cell. 4. Mitochondria is where the aerobic respiration reaction happens so the cell has energy. 5. Ribosomes is where protein synthesis happens and are on the rough endoplasmic reticulum. Organelles only in plants. 1. Chloroplast is where photosynthesis happens, and this gives food to the plant. It has chlorophyll pigments, which make the plant green by harvesting the light for photosynthesis. 2. Permanent vacuole is in the cytoplasm, and it's made of cell sap, which is a sugar-salt solution. This improves the cell's rigidity. 3. Cell wall is also found in algal cells, and it's made of cellulose and gives strength to the cell. Organelles in bacteria cells. 1. Cytoplasm. 2. Cell membrane. 3. Cell walls have similar function to the cell wall of plant cells, but they're not made of cellulose. Instead, they're made of peptidoglycan. 4. Chromosomal DNA is DNA that floats in the cytoplasm as bacteria cells have no nucleus. 5. Plasmids are small rings of DNA which code for extra genes. 6. Flagella is a long, thin tail which allows the bacteria to move. Specialized cells. Cells become specialized after being differentiated, which is when the cell gains new subcellular structures, so it's better suited to do its role. Animal cells only differentiate once, and this can either be early on or later like stem cells, whereas plant cells can differentiate many times in their lives. Specialized animal cells. 1. Sperm cells are specialized to carry male DNA to egg cell for reproduction. They have a tail which allows it to swim to the egg, and it has a lot of mitochondria which gives it energy by respiration, so it can move. Acrosomes are digestive enzymes in the head which allows it to break the outer layer of membrane on an egg cell. They have a hypoid nucleus, so it has 23 chromosomes and not 46 like the rest of the body cells. 2. Egg cells are specialized to accept one sperm cell and develop into an embryo. Surrounded by special cell membrane, which only accepts one sperm cell when fertilized and becomes impermeable after it accepted the sperm cell. Lots of mitochondria gives energy so the egg can develop into an embryo. Large in size and cytoplasm, so quick divisions as embryo grows. 3. Ciliated epithelial cells are specialized and have long, hair-like projections called cilia, which move bacteria trapped in sticky mucus down to the stomach where it's killed by stomach acid. This is how our body protects us against illness. Specialized plant cells. 1. Root hair cells are specialized to take up water by osmosis and mineral ions by active transport from soil. Root hairs are found on the tips of roots and gives the roots a large surface area so more water can move in. Large permanent vacuole affects speed of movement of water from soil to cell. Mitochondria give energy from respiration for active transport of mineral ions into root hair cells. 2. Xylem cells are specialized to transport water and mineral ions up the plant from roots to shoots. Chemical called lignin is released which causes cells to die, and it becomes hollow and cells are joined end to form a tube, so water and mineral ions can move through. Lignin is deposited in spirals which allow cells to withstand pressure from movement of water. 3. Phloem cells are specialized to carry product of photosynthesis to all parts of the plant. Cell walls of each cell form sieve plates when they break down, so substances can move from cell to cell. Companion cells have mitochondria, which gives phloem cells energy, so they can stay alive. Microscopy. Small structures like cells can't be seen without using a microscope, because microscopes enlarge the image. Light microscopes have two lenses. It produces light from the bottom. It has a magnification of around 2000, and magnification is how much times larger the microscopic image is compared to the actual image. It has a resolution of around 200 nanometers, and resolution is the ability to distinguish between 2 points that are close to each other. The lower the resolution means it's more detailed. When viewing a cell using light microscope, you can only see the nucleus, cell membrane, and cytoplasm. Electron microscopes allow you to see deep into cell, so you're able to see subcellular structures like mitochondria, ribosomes, chloroplast, and plasmids. Electron microscope views cells in more detail because electrons have a shorter wavelength than light waves. There's two types of electron microscopes, which are scanning electron microscopes and transmission electron microscopes. Scanning electron microscopes create a 3D image with a higher resolution than transmission electron microscopes, which produce a 2D image. Have a magnification of 2 million, and scanning electron microscopes have a resolution of 10 nanometers, and transmission electron microscopes have a resolution of 0.2 nanometers. Used to view dead cells as they're used in a vacuum. Electron microscopes allow organelles to be seen more clearly and detailed, especially very small organelles like ribosomes. Transmission electron microscopes are used to discover viruses as they're very small. Size and scale using microscopes. Calculate magnification using light microscope by multiplying magnification of eyepiece lens with magnification of objective lens. Calculate size of object by dividing the size of images by magnification. Represent very small decimal numbers or very large numbers using standard form. Number has to be between 1 and 10 and is multiplied by 10 to the power of a certain number when represented in standard form, like 5.0 multiplied by 10 to the power of 5 would be 500,000. The number you're using to represent the power is either negative for very small decimal numbers or positive for larger numbers. Cells and organelles are small and use order of magnitude to see how much bigger or smaller the image is compared to the actual size. Order of magnitude is 10 to the power of 1 if the image is 10 times bigger than the actual size of the cell. Measurements you need to know are: centi is 0.01, milli is 0.001, micro is 0.0000001, nano is 0.000000001, pico is 0.00000000001. Estimating is used when you're not sure what the exact number of something there is, or it takes way too long to count. For example, if there are 201 meter by 1 meter boxes filled with marbles, you would count the number of marbles in one box and multiply the number of marbles in that one box by 200 in order to get an estimate for the number of marbles in all the boxes. So, say there's 20 marbles in one box, then multiply 20 by 200 for 4,000, meaning that on estimate there's 4,000 marbles in all the 200 boxes. 4,000 marbles is just an estimate, so the real number will probably be different, and the more samples there means you will get closer to the real number. Enzymes. Enzymes are biological catalysts that speed up the rate of reaction without being used up. Enzymes are present in many reactions, so they can be controlled. They break up lathe molecules into small molecules. Shape of enzyme's active site is unique and complementary in shape to the substrate, so they can bind to each other and form an enzyme-substrate complex, and this allows substrate to be broken to form products which are released from the active site. Enzyme's specific shape to the substrate is called enzyme specificity. Enzymes need optimum pH and temperature, as it's a protein that denatures when the optimum conditions are not maintained. They also need an optimum substrate and enzyme concentration, but changes in these conditions won't make the enzyme denature. As temperature increases, so does the rate of reaction increase, but above the optimum temperature, the rate of reaction decreases and eventually stops. When it's too hot, the bonds that hold the enzyme together begin to break, and this changes the shape of the active site, so substrate can't bind to it. So enzyme-substrate complex can't form, and the products aren't formed from this reaction. Changes to enzyme's active site is when the enzyme denatures. Doesn't denature if temperature is too low, but the rate of reaction slows down. As pH increases or decreases, the rate of reaction decreases, as there's only one pH that the enzyme works optimally in. Based on where the enzyme is, this optimum pH level is different. So, in acidic environments, the optimum pH is lower, and in alkali environments, the optimum pH is higher, but it's normally neutral, so 7. Changes in pH cause bonds and enzyme to break, and this changes the shape of the active site, so the substrate can no longer bind to it. So no enzyme-substrate complexes are formed, and products also aren't formed. Enzymes become denatured. As the substrate concentration increases, so does the rate of reaction increase because the rate of enzyme-substrate complexes forming increases. But this only happens to a certain point called the saturation point, and after this point the rate of reaction stays the same. Saturation point's different for every enzyme. As the enzyme concentration increases, so does the rate of reaction increase because the rate of enzyme-substrate complexes forming increases, but this only happens to a certain point called the saturation point, and after this point the rate of reaction stays the same. Saturation point's different for every enzyme. Rate calculation for enzyme activity. Rate calculation used to find how fast an enzyme reacts, so basically the rate of reaction. Rate calculation calculated by dividing change by time. Change is change in substance, and time is time taken for change to happen. Rate is the way enzymes catalyze reaction, and change depends on temperature, pH, substrate concentration, and enzyme concentration. Enzymes as biological catalysts. Carbohydrates convert carbohydrates into sugars. An example of a carbohydrate is amylase, which breaks down the carbohydrate starch into the sugar maltose. Amylase found in saliva and pancreas. Protease converts proteins into amino acids. An example of a protease is pepsin, which breaks down proteins in amino acid. Pepsin found in stomach, pancreas, and small intestines. Lipase converts lipids into fatty acids and glycerol. Lipase in pancreas and small intestines. Products of these enzymes are soluble in bloodstream to be carried to all cells in our body. Products of enzymes used to build new carbohydrates, lipids, and proteins, and some glucose is used in respiration. Building carbohydrates, lipids, and proteins need different complex enzymes to increase rate of reaction. Transport in and out of cells. Substances like oxygen, glucose, and waste products are transported on and out of cells. Passive transport is transport that doesn't require energy. Substances are transported in 3 ways. 1. Diffusion is a type of passive transport, and molecules move randomly and collide with each other. Diffusion is the net transport of molecules from an area of high concentration to an area of low concentration. It's the net transport as most of the molecules move in that direction and some don't. 2. Osmosis is a type of passive transport, but only happens in water. Osmosis is when water molecules move from a dilute to a concentrated solution across a partially permeable membrane. 3. Active transport is a type of active transport, so requires energy from ATP produced from respiration. It requires energy as molecules are transported against the concentration gradient from an area of low concentration to an area of high concentration. Calorimetry. This measures energy taken in and out in a chemical reaction. This is used to measure calories in food. Steps to measure calories in food. Step 1. Place 50 milliliters of water into a test tube. Step 2. Find starting temperature of water. Step 3. Heat food and hold the test tube above the burning food sample at 45 degrees angle. Step 4. Measure final temperature of water. Step 5. Calculate energy transferred, joules, to water by multiplying the specific heat capacity, which is 4.2 with mass of water, grams, and temperature increase, Celsius. Practical variables. Control variables are things you're keeping the same throughout the practical. Dependent variables are measured during the experiment. Independent variables are the ones that are being changed throughout the practical. Core practical. Investigating biological specimens using microscopes, including magnification calculations and labeled specific drawings from observation. Microscopes are important when looking at biological specimens. Parts of a microscope. Eyepiece is where we look through to see the specimen. Stage is where the specimen is the part of the microscope that has the specimen on. Lens, increase magnification of a specimen. Focus will sharpens the image. Objective wheel for magnification. Use microscope in following way. 1. Place slide on stage and look through eyepiece lens. 2. Turn focus wheel to get a better resolution. 3. Start with lowest objective magnification. 4. Increase magnification slowly and refocus after changing magnification. Prepare specimen. 1. Get a thin layer of cells from the sample by peeling them off or using cotton bud. 2. Add small amount of chemical stain like iodine so specimen is more visible and seen in more detail. 3. Put the thin layer onto the slide. 4. Lower cover slip onto the sample at an angle to avoid air bubbles. 5. Then lightly dab it with a cotton bud. Core practical. Investigating the effect of pH on enzyme activity. Enzyme used is amylase, which breaks down starch into sugars like maltose. Iodine is used to test for starch. When starch is present, the color changes from orange to blue-black. Amylase has optimal pH, and use this experiment to estimate what is. 1. Put 50 milliliters of water into a test tube and label it with the pH that the test is going to be done at. 2. Place it a Bunsen burner to keep the solution at a constant temperature so this doesn't affect results. Temperature is a control variable. 3. Put 2 centimeters cube of amylase solution, 2 centimeters cube of starch, and 1 centimeters cube of pH buffer solution in a test tube and start a stopwatch for 10 seconds. 4. Then place a drop of the solution into one of the wells which has iodine in it. 5. If the mixture turns blue-black, it shows that starch hasn't been broken down yet. 6. Repeat for another 10 seconds and test it again as shown in step 4. Do this until the solution doesn't change colors and record the time it takes. 7. Record results on graph, which has pH on x-axis and time taken for reaction to complete on y-axis. Control variable is mass of water. Dependent variable is time taken to see color change. Independent variable is the different pHs. Core practicals. Osmosis in potatoes. Measure water gain by potatoes for osmosis practical. 1. Cut potato into small discs which are the same size. 2. Blot potato to remove excess water. 3. Measure mass at the start. 4. Place disc in sucrose that have a different concentration. 5. Blot with tissue and then record mass. 6. Find difference in mass by subtracting start mass from final mass. 7. Use percentage change calculation to find gain or loss of mass. Percentage change equation is change in mass divided by start mass multiplied by 100. Control variable is the type of potato. Dependent variable is mass of potato. Independent variables is sucrose concentration. Thank you for watching. Please like, comment, and subscribe.